Today we are going to discuss about noble metal nanoparticles, especially the synthesis and application of nanoparticles of gold, silver, rhodium, palladium and platinum. This is from the chapter Nanomaterials, semester 4 of MSc Chemistry. And what are the unique properties and applications of noble metal nanoparticles? Noble metal nanoparticles with controlled size and shapes have increased scientific interest and technological significance owing to their unique physical and chemical properties, and they are used in wide range of applications that include catalysis, photonics, sensing, energy conversion and storage, and especially in biomedicine. The tunable size and shapes of noble metal nanoparticles would determine their catalytic behavior in terms of activity, selectivity, and durability through the adjustment of number and geometry structure of the active sites. The high activity is associated with large surface to area volume ratio induced by small particle size. And now we will see one by one. First of all, the synthesis and application of nanoparticles of gold, and this is taken from these two journals. As we all know, the properties of gold nanoparticles are different from that of the bulk because bulk gold is yellow solid, and we can see this in the figure, and it is inert in nature, while the gold nanoparticles are wine red solution. You can see the 20 nanometer range wine red solution of gold nanoparticles, and they have an excellent antioxidant property. The inert particle interparticles interacts, and assembly of gold nanoparticles play a key role in the determination of properties and these nanoparticles. Gold nanoparticles exhibit various size range. That is, they have a size range of 1 nanometer to 8 micrometer, and they exhibit several shapes such as they show nanosphere shape, nanoshell, branched, nanocube, nanostar, nanorods, nanoclusters. And about the synthesis, there are many methods of synthesis. There are physical method of synthesis, chemical synthesis, and green synthesis methods. And we are discussing the two of the methods, that is the chemical and green synthesis. In chemical methods, also there are large number of synthesis possibilities. But first of all, we will discuss the most simplest one, and this synthesis was proposed in 1951. Gold nanoparticles was prepared by the treating hydrogen tetrachloroiodate with citric acid in boiling water. When the citric acts as both reducing and stabilizing agent, the citric acid here is acting as a reducing and stabilizing agent. Moderately stable spherical AU nanoparticles with diameters of 10 to 20 nanometers were found. Though larger nanoparticles, example 100 nanoparticles, can also be prepared in this method. These citrate stabilized gold nanoparticles can undergo irreversible aggregation during functionalization process with the thiolite decants. This is the simplest of the chemical synthesis. And when it comes to green methods, the green chemistry synthesis routes are environmentally friendly and non-toxic. A facile green biosynthesis method for the preparation of gold nanoparticles of size 25 plus 7 plus or minus 7, it's plus or minus 7 nanometers, was reported by using eggshell membranes. You can see that they're using eggshell membranes, how the gold nanoparticles was prepared. In this method, ESM, that is the eggshell membrane, was immersed in the solution of hydrogen tetrachloride without using any reductant. We are not using any reductant. We are only using the eggshell membrane here. And another green approach, and so the gold nanoparticles were prepared. Another green approach was developed by this developed to synthesize gold nanoparticles of a range 5 to 17 nanometers by using high power ultrasounds. These are the two methods of synthesis of gold nanoparticles. And when it comes to application, there are immense applications for gold nanoparticles. We can search the web, and otherwise you can refer the papers given. Gold nanoparticles have unique electric and magnetic properties due to their shape and size, and they have been received great attention in research area, especially in the field of biological tagging, chemical and biological sensing, optoelectronics, photothermal therapy, biomedical imaging, DNA labeling, microscopy, and photoacoustic imaging, surface enhanced diamond spectroscopy, tracking and drug delivery, catalysis, cancer therapy, etc. Gold nanoparticles based sensors can detect various metal ions by working on the principle of the color change due to the aggregation of gold nanoparticles. Such type of sensors have been widely used in the detection of copper, mercury, lead, and arsenic in water, and the list goes on. We have already discussed there are many shapes of gold nanoparticles, and especially the one gold nanorods have gained much attention in recent past years due to their specific optical and chemical properties and hence used for biological applications. And these gold nanoparticles with unique anisotropic geometry enables them to get tunable absorption in both visible and near IR, that is NIR regions, and make them suitable for potential applications in the field of biosensing, gene delivery, and photothermal therapy. And another application of the gold nanoparticle is it is used in X-ray CT imaging as molecular probes. Earlier, iodine-based agents were used, and gold nanoparticles are seen to be more advantage than the conventional iodine-based agents, as gold has higher absorption coefficient than iodine due to the higher atomic number and electron density. So it enhances the CT contrast more than iodine. Also, another advantage is non-cytotoxicity. Cytotoxicity means the toxicity towards the cell, and these gold nanoparticles has no toxicity towards the cell. And important benefit is regarding the surfaces, as they have large surface area due to which their surfaces are readily available for modification with target molecules or specific biomarkers and application in biomedical purposes. Also, the gold role of gold nanoparticles in biological sciences is very important because of its compactability, conjunction, and these particles to the biomolecules and their tunable optical properties, which are due to the shape, size, and surface area of the nanoparticles. And they have a small size, large surface area, and shape, and crystallinity. Gold nanoparticles are proved to be excellent therapeutic agents. They can easily travel into the targeted cells and can bear high drug loading so that they can act as drug loading vehicles. Also, as we have already mentioned, they are used as therapeutic agents or vaccine carriers. Also, their epidermal delivery of DNA vaccines. They are the best delivery vehicles for DNA vaccines. The coated walls of gold nanocages with the temperature sensitive polymers were used as drug carriers, which releases their effectors with interaction of near infrared radiations. Gold nanoparticles with a small size and diameter about 1 nanometer are able to cross the cell membranes and nucleus to interact with the DNA. So these all make them a special drug delivery vehicle or drug delivery agent. The gold nanoparticles having strong affinity for alkenes as compared to the other transition metal catalysts, but the homogeneous system are not favored economically and environmentally because the rapid reduction of active gold complexes into inert metallic gold during the CH alkane activation. And they have strong affinity, but because of their homogeneous system, it is not favored. Due to their unique optical and electrical properties, the gold nanoparticles have been widely used for color indicating probes of various in the way development of analytical techniques which are used of sensing of various analytes. As we have already seen, it is used to detect many metals, and when they interact with metal the color changes, and this is in how we can sense these metals, especially in water. And another applications are the noble metals such as gold nanoparticles key role in biosensors, especially because of their large surface area, small size, good biocompatibility, conductivity, and excellent catalytic properties. Nanoparticles combined with the biomolecules have been used in biosensing applications. It was used in biosensing, especially in detecting glucose concentration with a limit of 0.025 millimolars. We can detect the glucose levels at these low concentrations also using gold nanoparticle, especially when the gold nanoparticles combine with the mecha MPS. And the most of the biosensors depend on this special kind of phenomenon called localized surface plasmon resonance of gold nanoparticles. So, in order to enhance the efficiency of biosensors, gold nanoparticles have been immobilized in solid surface. We will discuss that what is meant by the surface plasmon resonance. Especially when it comes to the noble metals, there is a phenomenon called as plasmon wave, and it is an optical technique utilized for detection of molecular interaction, especially when something binds into the surface. The change in plasmon waves can be detected and studied, and how will they interact with the substrate can be analyzed. Binding of mobile molecules to a molecule immobilized on a thin metal surface, that is here, it is the ligand, changes the refractive index of the film. SPR is the basis of many standard tools for measuring absorption of materials onto planar metal, especially gold or silver. We will deal with this technique in silver when it comes to silver nanoparticles also surfaces or onto the surface of the metal nanoparticles. It is the fundamental principle behind many color-based biosensor applications, different lab on a chip sensors and diatom photosynthesis. The lab on a chip we will discuss lately, and these are all based on the surface plasmon resonance. And the functionalized gold nanoparticles with calcium derivative, crown ethers, DNA, and peptides has been used in the sensing applications, mainly in the recognition of amino acids, quaternary ammonium salts, and pyridinium. And these are all the applications, the sensing applications which the uh, gold nanoparticles possess. Indium titanium oxide electrode modified with gold nanoparticles or titanium dioxide composites have been used in the estimation of catechol hydroquinone with the help of voltammetric methods. The catechol has been successfully determined in the tree samples using this method. Adopting this method. Another important application of the gold is it is used as a memory device. The gold nanoparticle coated with suitable insulator exhibit excellent stability for memory devices, and this insulation helps in prevention of accumulation of charges when the applied field has been removed. So a memory field, the application in memory field is a hot area of research now. This we have discussed earlier, surface plasmon resonance, and the we will discuss about the drug delivery of gold nanoparticle. The targeted drug delivery, why is it used than the conventional drug therapy? Because of the drug target, the main area is only affected, and because of it, uh, the minimum side effect is caused than the conventional drugs. The primary goal of the developing anti-cancer agent is to minimize the various side effects caused by conventional drugs and to improve the selectivity and efficiency of drugs. Gold nanoparticles has the ability of bioimaging of the affected cancerous cells for therapy. The effective drug delivery system or drug therapy is important to investigate about the biological effects of the nanoparticles, as gold nanoparticles have unique physical and chemical properties and the strong binding attractions for thiols, proteins, carboxylic acids, and disulfates. So they have been extensively used in the field of biosciences, especially drug delivery in cancer therapy. And the gold nanoparticles follow three pathways, especially the receptor media endocytosis, phagocytosis, and the fluid phase endocytosis. These are the three main pathways the cellular impact take on. And the toxicity of the gold nanoparticles depends on the shape, size, synthesis method, surface charge, surface coating, and functionalized molecules. But overall cytotoxicity of gold nanoparticle is in the acceptable level. That is the important thing, as gold nanoparticles are considered to be non-toxic agents. The factors like drug delivery and transport. There are two main factors: drug release and transport, which is very important for the efficient drug delivery system. Drugs are loaded on the nanocarriers by non-covalent interactions or through covalent conjugation with the help of prodrugs, which is treated by the cell. The gold nanoparticles have functional stability, flexibility due to their monolayers, so they prove to be an efficient system. And the research is going on for the drug delivery system. And next, we will move on to silver nanoparticles. As we all know, silver has the unique properties. It has an antibacterial, anti-fungicide, and antiviral properties. And silver draws tremendous actions towards the academic and industrial interest. Besides of their benefits of their biomedical applications, when the size of the silver particles decreases to nanoscale, the high surface area and surface energy make silver one of the best candidates for catalysis and optical absorption. Here also, the surface plasmon resonance play an important role when placed in a dielectric environment, leading to the higher optical absorption efficiency. Therefore, the applications of silver nanoparticles expanded from biology, electronics, textile industry, and catalysis to optical and photovoltaic materials such as solar cells. Nowadays, it is also investigated in solar cells and other photoelectric materials. Fibers with nanostructures have been intensively investigated because of their high surface to volume ratio. Compared to the traditional materials, membranes and films composed of nanofibers have extremely high surface area and a nanometer grade micropores, which have provide both larger number of active reactive sites and for chemicals and excellent filtration properties and breathability when being used as insulating fabrics and biomaterials such as wound dressing and protective textiles. Nanomaterial based on silver can synthesized by chemical physical methods with the presence of reducing agents or laser ablation. And these points are taken from this research journal, these two journals. The DOI number are given. And when it comes to synthesis, we will first discuss about the chemical synthesis. The chemical synthesis, in chemical synthesis, we use the chemical reduction, the most employed way to prepare silver nanoparticles as stable colloidal dispersion in water and other organic solvents. Many studies have shown that after the silver ions are reduced to silver atoms, they will further agglomerate or oligomeric clusters into oligomeric clusters, leading to the formation of silver nanoparticles. The main precursor we use is silver nitrate, silver acetate, silver citrate, and silver chloride. And the major reducing agents are borohydrate, citrate, ascorbate, and compounds with hydroxyl or carboxyl groups such as alcohol, aldehydes, carbohydrates, and their derivative. It has been shown that the growth mechanism of silver nanoparticles is strongly influenced by the properties of this reducing agent, especially when we are using the stronger reducer, such as the borohydrates, generates monodisperse silver nanoparticles with a larger size, while the mild reagents such as ascorbate and citrates create smaller number of nuclei at slow rates, which in turn promotes the generation of silver nanoparticles with smaller size and wider dispersion. The advantage of borohydrate is it's monodisperse, and the disadvantage of ascorbate and citrate is wider dispersion. Also, it is also interesting to mention that the dispersion media plays an important role in particle sizes and morphology of silver nanoparticles during reduction. The dispersion media, which refers to the solution and solvent systems, serves as protective agents which can absorb or bind onto the particle surface to avoid the agglomeration of the particle. And the major stabilizing agents or protective agents are the polymers such as PVP, PAA, PMAA, that's the polymethyl acrylate, and the polymethyl acrylic acid, polyethylene glycol, and such. So, and the greener approach is in this method, biosynthetic methods employing natural occurring reducing agents such as polysaccharides, biological microorganisms such as bacteria and fungus, or plant extracts, that is green chemistry, has emerged as a simple and viable alternate to more complex chemical synthetic procedure to obtain AG nanoparticles. There are three major steps in green chemistry perspectives and the green approach, that is the selection of the solvent medium, selection of the reducing agent, and the selection of non-toxic substances for the nanoparticle stability. Especially one of the preparation method is using synthesis of starch Ag nanoparticles, where starch is used as a capping agent and D-glucose as the reducing agent in a heating system, generally heating system. Here, the starch solution mixture avoids the use of relatively toxic organic solvent. Additionally, the binding interaction between the starch and Ag nanoparticles are weak and can be reversible at high temperature, allowing separation of the synthesized particle. Especially, this is the thing, the capping agent starch and the D-glucose. If we are using these two things in the precursor of either the silver nitrate, silver acetate, silver citrate, or silver chloride, this is called to be as a green approach. And these are much many applications. There are many applications for silver nanoparticles, especially in textiles, biomedical application, that includes cancer therapy, diagnosis, drug delivery, selling, imaging, etc. Healthcare, that's the UV protection, oilments, it is used as in oilments, food, agriculture, especially in packing, and catalysis, the fuel cell catalyst, and environmental, especially in the wastewater treatment. And when it comes to application, we all know, we have already discussed, because of this antibacterial agent, it is used in health industry, food storage, textile coating, and a number of environmental applications. As antibacterial agents, it is also used in disinfecting medical devices and home appliances to water treatment. Moreover, this encourages the textile industry to use silver nanoparticles in different textile fabrics. Silver nano composite fibers were prepared containing silver nanoparticles incorporated inside the fabric. That's an excellent innovation. And the cotton fibers containing Ag nanoparticle exhibited high antibacterial activity against E. coli bacteria. And the electrochemical properties of Ag nanoparticles incorporated with the nanoscale sensors that can offer fast response times and lower detection limits. The electrodeposited Ag nanoparticles onto aluminum plates, gold micro-patterned electrons that showed a high sensitivity to hydrogen peroxide. That is, we can detect this hydrogen peroxide even at lower concentrations. The catalytic properties of nanoparticles differ from the chemical properties of the pulp. For instance, bleaching of organic dyes by applications of potassium peroxodisulfate in aqueous solution at room temperature is enhanced strongly by the applications of silver containing nanoparticles. Also, it catalyzes the chemiluminescence from luminol hydrogen peroxide system with catalytic activity better than AU and platinum colloid. And especially when it comes to water filtration and treatment, we all know, because of the silver's antibacterial property, we are using it in water treatment. There are other antimicrobial agents such as zinc oxide, MgO, and TiO2. But silver nanoparticles are shown the most effective, to be reported as the most effective antimicrobial agent. When pairing with hydrogen bonded multi layers assembled on magnetic microspheres, it can be delivered and localized in specific regions without contaminating the surroundings by using magnetic fields. When it is used as a composite, use with Fe3O4 at the silver nanoparticles, both with both super paramagnetic and antibacterial property. We know that silver nanoparticles has this antibacterial property and Fe3O4 has the super paramagnetic property. And when we form a composite using both these materials, it has shown tremendous properties against bacteria such as E. coli, Staphylococcus epidermidis, and Bacillus stabilis. In addition to the benefit of paramagnetism, which allows the material to be easily removed from water, mesoporous polymer nanofiber membranes can be designed with specific pore sizes and desired filtration properties to enhance the water treatment efficiency and recyclability. With the antibacterial property, these bacteria can be removed, and because of this super paramagnetism, the material, especially the composite, can be removed from the wastewater treatment. That is one of the major important thing. The nanocomposites of superparamagnets or silver nanoparticles or polymer nanofibers can be a promising water disinfectant. And the another thing is it is used in nano paints, especially antimicrobial nano paints. We are seeing um, all these things now, and the silver nanoparticles in all these things in advertisement, especially Asian paints or something. We are seeing it, the silver nanoparticles in silver nanoparticle or polymer nanocomposites are highly stable due to the polymer matrix. They can be sustained up to 200 degrees Celsius without significant oxidation and aggregation, which enables them to produce production of silver nanoparticle embedded homogeneous paints. In the application of silver nanoparticles or polymer nanopaints, the silver nanoparticles are synthesized in polymer solution with one-step method following by drying process, and the resultant silver nanoparticle embedded drying oil is an excellent coating material and can be applied on various surfaces including wood, glass, and polystyrene. The surface coated with the nanopaints exhibited outstanding antibacterial properties by killing both gram-positive human pathogen and gram-negative bacteria. And that is all for silver nanoparticles, and we will move to platinum nanoparticles. We all know that the platinum is one of the rarest and most expensive of the metals, and the platinum nanoparticles are trustworthy scientific tool that are being explored in various biotechnological, nanomedical, and pharmaceutical pharmacological fields. They are unique because of their large surface area and their numerous catalytic applications, such as they are used in automotive catalytic converters and petrochemical cracking catalysts. The platinum nanoparticles has been widely utilized not only in industry but also in medicine and diagnostics. Platinum nanoparticle extensively studied because of their antimicrobial, antioxidant, and anti-cancer properties. We all know the what we are learned from cisplatin is one of the first anti-cancer drug scientists have made, and the synthesis method. First, we will discuss about the chemical approach and the green approach lately in the next slide. And the platinum nanoparticles has been prepared by reducing hexachloroplatinate. A solution of hexachloroplatinate is rapidly stirred upon the addition of reducing agents such as hydrogen gas, which in turn causes platinum ion to be reduced into neutral platinum atoms. The solution rapidly becomes supersaturated due to the formation of large number of platinum atoms. This process is followed by precipitation of platinum to nanoparticles. It's a simple method, and the particles attain uniform size and shape in the solution is stirred vigorously. The aggregation of platinum nanoparticles can be prevented by adding stabilizing agents such as sodium polyacrylate uh, to the solution. And about the green approach, this is a synthetic route given. We will have a plant extract, and we will filtrate the plant extract, and keep upon stirring we will add hexachloroplatinate to it, and upon centrifugation we will get the platinum nanoparticles. The green synthesis of platinum nanoparticles is using plant extracts. The plant-based synthesis of platinum nanoparticles have numerous advantages over other types of biological methods. The first synthesized nanoparticles in living plants and fabricated gold nanoparticles from alfalfa seedlings with the size range from 2 to 20 nanometers. And their application includes the electrocatalysis and catalytic converters. It is used as electrocatalysis and catalytic converter, magnetic nanopowders, polymer membranes can used in cancer therapy, coating plastic nanofibers, and in textile industries. These are some of the applications listed, especially applications that is used in biomedical field, the photothermal therapy and radiotherapy. The platinum nano, as we discussed, is used in anti-cancer drugs, used in biomedical imaging, anti-inflammatory, nanodiagnostics, antibacterial, antifungal, and at last in drug delivery. We know the story of how the cisplatin and the anti-cancer activity of platinum, and I will not discuss. And the next application is glucose detection application. Especially we were when we were looking into the gold nanoparticle, we can also see the glucose detection there. Enzymatic glucose sensors have drawbacks that originates from its nature. The non-enzymatic glucose sensors based on platinum-based electrocatalysts offer several advantages, including high stability and ease of fabrication. Many novel platinum and binary platinum-based nanoparticles has been developed to overcome the challenges of glucose oxidant on the surface, such as low selectivity, poor sensitivity, and poisoning in the interfacing species. These are the drawbacks which have, but using platinum, these drawbacks can be avoided. And it is also used as hydrogen fuel cells. Among the precious metals, platinum is the most active towards hydrogen oxidation reaction that occurs in the anode of the hydrogen fuel cells. Okay, this is all for platinum. And when it comes to palladium, palladium nanoparticles are of great importance as catalytic materials as well as for a number of other applications such as hydrogen storage and sensing. This transition metal occurs as a dry, stable powder with silver white in color, and it is ductile in nature. Palladium is the lightest metal in the platinum group and resist oxidation. This is very important thing. Resist oxidation under standard temperature conditions. In addition, it has the ability to absorb substantial amount of hydrogen. And when it comes to abundance, palladium within the Earth crust has 0.015 ppm by weight, which is the threefold than that of the platinum. But the price of palladium is above the acceptable levels for large scale commercial application. And it is feasible that we will the platinum and palladium has the same properties, and we can use palladium instead of platinum because platinum resources are very cheap in nature and palladium is more abundant in nature, and it is a one of the major sustainable thing we can do. Though the use of dye synthesis technique, PD based nanoparticles have potential to provide superior and cost-effective solutions to meet the requirements of present and evolving electrochemical applications, specifically the development of hydrogen economy. Palladium is used mainly in jewelry, but also as a catalyst in special alloys also. Palladium catalysts are typically used to break down petroleum and high quality gasoline and other products. This is one of the major important thing of palladium. In addition, it is used to obtain essential chemicals such as sulfuric acid, which is termed as the king of chemicals, which in turn is used in the production of paper, fabrics, and car batteries, for example. Some catalytic converters used in automobiles also use palladium catalyst to help the fuel burn more efficiently. Palladium is frequently alloyed with gold, silver, and copper. As expected, the properties of the resulting mixture differ from that of the individual compounds. Alloy is used in various varieties of precision products such as ball bearings, springs, watch balance, wheel, uh, surgical instruments, electrical contacts, and astronomical mirrors. Palladium alloys are also used widely in the industry. Palladium plays another important role. We all know that it is used as a catalyst in many reactions such as Higgs, Suzuki, and Stille coupling reactions. And in addition, platinum nanoparticles. This is especially the points related to platinum, and these 2 points are applications of platinum nanoparticles. Show high catalytic activity for ethanol electro-oxidation in basic medium and have the potential to be used as the surface enhanced Raman scattering substrate where the SERS enhancement intensity has been shown to the size and morphology dependent. Thus, the development of new synthetic methods for the formation of PD nanoparticles with well controlled size and shape is important for the large number of future applications. This is one of the most important synthesis of palladium nanoparticles, especially as we can see, this is a hydrothermal method using the autoclave. And the advantages of hydrothermal methods, as we have studied earlier, it is low cost, facile, and temperature templateless approach. And this is how we are making these palladium based nanoparticles. And it was proposed by Adams et al. And my co-workers prepared PDCD nanoparticles, that is the palladium and cadmium nanoparticles, which were grown directly on titanium plates with a facile hydrothermal reduction method. The etched titanium substrate were introduced into Teflon line autoclaves along with the following. We used 5 millimolar PDCl2, 5 millimolar cadmium nitrate, and 1 molar ammonium formate up to a volume of 10 milliliter. Varied concentration of cadmium nitrate precursors were added to obtain the desired PD-CD ratio, and the autoclave was heated to 180 degrees Celsius for 2 hours, and once cooled, the PD-CD coated plates were annealed to 250 degrees Celsius for 2 hours in the tube furnace under an argon flow to ensure good surface adhesion of CD, PD thin films, and this resulted in the nanomaterials formed alloyed PDCD dendritic structures. And next, we are going to discuss about the synthesis and application of rhodium nanoparticles. Rhodium nanoparticles has many advantages like applications like CO reduction. It is used in CO reduction, NO reduction, hydrogenation, hydroformylation, electrooxidation. And given the rapid growing the interest of plasmonically assisted photocatalytic process, rhodium represents a tremendously promising metal for combining plasmonics and catalysis. UV plasmonic properties of rhodium nanoparticles has been by means of surface enhanced Raman spectroscopy, surface enhanced and charge transfer enhanced fluorescence, and photo induced degradation. Furthermore, rhodium-based catalysts are especially stable for use under harsh reaction conditions because of its strong resistance to the etching of acid or base and heat. This is one of the major chemical property, superior property that rhodium has compared to other noble metals. They are more superior and more strongly resistant to etching of acid and base and towards heat. This is a synthetic method that we are using. The typical synthesis, it's a chemical synthetic route. The ethylene glycol was placed in a three-neck flask equipped with the reflex condenser and a magnetic stirrer bar and heated to air at a designated temperature for 1.5 hours. In a separate vial, Na3RHCl6 and PVP polyvinyl pyridine was were each dissolved in Eg, that is the ethylene glycol at room temperature. The molar ratio of Na3RHCl6 to the repeating unit of PVV was controlled at 1 is to 5, and the two solutions in Eg were injected simultaneously into the heated Eg using a syringe pump at the rate of 0.2 milliliter per minute for the formation of rhodium nanoparticle. Combining microwave radiation with the piol reduction can be a typical strategy. This is the first method, and the second method is the first ethylene glycol method plus the microwave irradiation method. Compared to the conventional heating ways, microwave heating, assisted heating resulted in more uniform nucleation and shorter crystallization time, which is conducive to the formation of uniform metal colloids. And the two methods are discussed. First is the simple heating method, and second is the conventional heating plus the microwave assistance method. And compared to the conventional heating, the microwave assisted method is more faster and uniform nucleation and for uniform nucleation. Extensive studies on formation of rhodium nanoparticles reveal fundamental difference during the nucleation phase that is directly dependent on the heating method. Microwave irradiation was found to provide uniform seeds for subsequent growth and larger nanostructures of desired size and surfaces. The nanoparticle growth kinetics is also markedly different from under microwave heating. While conventional heating generates yields particles with a mixed morphology, microwave synthesis constantly provides a majority of tetrahedral particles at intermediate size between 5 to 7 nanometers or larger cubes more than 8 nanometers upon further growth. The high resolution uh, transmission electron microscopy, that is the HRTEM, indicates that RH seeds and larger nanoparticles obtained from microwave assisted synthesis are more highly crystalline and fisted versus the conventional prepared counterparts. The microwave prepared RH nanoparticles also show apparently twice the catalytic activity of similar size conventionally prepared nanoparticles as demonstrated in the wafer phase hydrogenation of cyclohexane. And these are the synthetic advantages the microwave irradiation assisted method has. They are more crystalline, they are more homogeneous, that's the uniformity is there, and the catalytic activity is also doubled for microwave assisted method than the conventional used method, and it is demonstrated using the vapor phase hydrogenation of cyclohexane. And these are um, the things associated with noble metal nanoparticles, and there are five of them: gold, silver, palladium, platinum, and rhodium nanoparticles. Okay, thank you.